Welcome to this video on architecting pregnancy safety studies, a structural blueprint for designing post-approval evidence ecosystems. Pregnant patients are actively excluded from pre-approval clinical trials. As a result, drugs enter the market with limited safety data for this population. Section 505(03) of the FD&C Act mandates post-approval safety studies when potential risks emerge. Safety data collection must span three distinct clinical windows: pre-pregnancy, during pregnancy, and postpartum and lactation. Each window tracks different outcomes, from conception rates to infant safety via breast milk. Three complementary approaches build a complete safety profile: case reports and series, pregnancy registries, and complementary studies. The FDA establishes no hierarchy of evidence among these methods, as each contributes uniquely to the overall safety assessment. Informative case reports act as the front line for rare signal detection. Key information includes detailed exposure, gestational timing, maternal history, external factors, and clinical evaluation. Pregnancy registries offer prospective rigor, minimizing recall bias and capturing real-world outcomes. However, they face severe operational gravity, including difficulties in recruitment and susceptibility to selection bias. Designing the registry cohort requires careful attention to the study population, capturing both prospective and retrospective enrollments. Establishing comparators, such as internal concurrent unexposed cohorts, is crucial to isolate drug effects. Registry operations involve a push-pull ecosystem for recruitment and retention. Recruitment is initiated by providers, facilities, and patients. Retention mechanics involve providing interim data reports and fostering longitudinal relationships. Analytics involve adjudicating outcomes and adjusting for statistical reality. Major congenital malformations must be classified by blinded experts. Sample size and data presentation must account for the rarity of outcomes and stratify by gestational timing. Complementary studies leverage secondary data, such as electronic health data, to validate and scale safety signals. They offer the power of massive sample sizes to quantify associations between exposures and specific outcomes. The mother-infant linkage model dictates EHD viability. The linkage bridge relies on matching identifiers. However, if a newborn is placed on a different health insurance policy, the exposure to outcome data pathway can be completely severed. Navigating live birth bias is critical, as EHD easily identifies live births but misses severe teratogenic defects, resulting in stillbirth or termination. Exposure ascertainment is also a challenge, since pharmacy codes record dispensing, not consumption. Filtering confounding variables isolates the true exposure effect. This involves three filters: confounding by indication, time-varying covariates, and recall bias in case control studies. Data sources for complementary studies include administrative claims, electronic health records, and population-based surveillance. Each offers unique strengths and limitations, such as sample size versus clinical depth and data adjudication. True regulatory confidence emerges only at the intersection of methodologies. Case reports detect signals, registries characterize risk, and complementary studies validate findings. No single study design is sufficient for a comprehensive safety profile.